Hey everybody, it's Miss Larson just here to explain the DNA discussion. So your job here is going to be to make a DNA model. You can click here to see this video, of course, which you would have figured out by now, and you'll be able to see an example down below. So for this discussion, you should show your knowledge of DNA by creating a model of DNA. Your model may be 2D, which means two-dimensional, or 3D, which is three-dimensional. You may draw the model using Google Drawing or on paper, or you may make it out of nearly anything. Legos, food, candy, clay, pipe cleaners, like you can do whatever you want. I've seen a lot of students get really creative with this in the past. You must identify and label several things in your model: adenine, thymine, guanine, and cytosine, a sugar phosphate backbone, and a double helix. But you are not limited to that. You can actually label more than that if you would like to, and you'll see that in the example. How can you share your work? Well, you can take a picture of your work and upload it to Canvas. You can make a video in which you show off your work and upload it to Canvas. You can take a picture and email it directly to me. Or if you are hybrid, you can turn your work in directly to me. Here's some little instructions on how to upload a video and how to upload a picture. Now, I want you to realize this. This is really important right here. Please note, I am looking for your original work on this assignment. You may use Google as inspiration, but you must create your own model. If you turn in a picture that you found on Google, you'll get a 0 for this assignment. Here's an example. You can make yours similar to mine. That would be fine. But here's my example, a DNA model. I got a human cell just to show that DNA is in the nucleus. It's wound up in chromosomes. Those chromosomes are made up of genes, which are just a section of chromosome. And you can see the shape of DNA is a double helix. Rosalind Franklin came up with that idea. And it's a twisted staircase shape. Now, when you look closely here, you can see the sugar phosphate backbone. You have a deoxyribose sugar and a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, a phosphate. And between the sugars are nucleotides, which is our monomer of the nucleic acid type of macromolecule. So that's kind of cool. But this is adenine and thymine, and they always bond together. And this is guanine and cytosine, and they always bond together. And you can see that there in the picture. So you make your own model. You can draw it. You can make a 3D model, do whatever you want to do. But make sure that you include and label these things: adenine, thymine, guanine, cytosine, a sugar phosphate backbone, and that double helix shape. Like I said, and like you can see in my example, you may include more, but you have to at least include these things. Thanks for listening.